So, things to know from here. Know the difference between pyrimidine and purine bases. I will ask about that. You don't need to know the formulas. You do need to know what base is pyrimidine, what is purine. Understand that in DNA the sugar is deoxyribose. Be able to tell apart nucleoside and nucleotide. Okay? It's one. Sorry, two. Understand what forms the backbone of DNA molecule. Phosphate and sugar. And understand the linkage that the phosphate group, shown here, connects 5' and 3' carbons of two different but adjacent sugars. Understand that this orientation gives DNA molecule directionality, which is essential for reading it in a certain direction. Okay? Recording and reading of genetic information. Go ahead.
And just to be clear, you said DNA was read 5' and 3', RNA is read.
So let me rephrase the question, okay? So you're asking about reading. DNA is always read 3 to 5', no exceptions. When RNA is read during translation, it is read 5 to 3. Now here, strands anti-parallel, which means, well, next slide will explain what it means. So if you look at this image on the right, okay, so this is 5' end, this is 3' end, so in some way this strand goes from top to bottom. The second strand in this image goes from bottom to top. Okay, can you see that? All right, so that means they are anti-parallel. Understand the roles of major and minor grooves. The role of this are just protein binding, okay? Obviously, base pairing, folks, ATGC at this point should be like burned into your memory, right? And understand that AT pair is weaker than GC pair because it has only 2 hydrogen bonds. Okay, whatever. Does that make sense? Because GC pair has 3 bonds. Okay? Now, because we have such a strict pairing between AT and GNC, in the DNA molecule that is double-stranded, the percentages of A and C are equal. The percentages of G and C are equal to, that's the Chargaff rule. So first of all, the illustration. So imagine you have a double-stranded DNA molecule and somehow you established that 20% of all bases are A. What's going to be the percentage of T bases? Also 20. What do we have left total for G and C? 60. 60. 100, right? Minus 20, I have 60. So G is going to be how many? 30, and C is going to be 30 as well. Does that make sense? This rule can be applied only to double-stranded DNA molecule. Okay. Not a lot of information here. Just understand that strands can be separated. You can break down those hydrogen bonds. And that is essential for reading the information because the information is written in those bases. This process of strand separation is called denaturation. So, what about RNA? Now, there are a lot of exceptions in microbiology in particular and biology in general. We aren't focusing on the exceptions. We aren't focusing on rare occasions. So for our course, differences between DNA and RNA that we should remember. First of all, this, ribose instead of deoxyribose, different sugars. Second, uracil against thymine. Okay, so there, if you see the sequence that contains U instead of T, this is RNA. Third, RNA is usually single-stranded, but it can form intramolecular base pairing. The structure here is called hairpin loop. Okay. Now, regarding this table, I will absolutely 100% ask you to match the type of RNA. I'm going to tell you specifically, and its function. Am I making sense? What is the function of mRNA? And you pick one of four. Or which of the following RNA types is responsible for transporting amino acids to the active site of the ribosome? And you tell me tRNA, yes. Making sense so far? Good? Okay, that's pretty straightforward. This, genome sizes. Again, just the order. Are there exceptions? Of course. Do we care? No. Am I clear? Bless you. Am I clear? Right. So we mentioned before that phenotype is determined by genes that are expressed at the given moment. Keep it in mind. Okay? I expect you knowing this. It's called central dogma of molecular biology, the order of events in the gene expression. You absolutely must know that. Okay? And to understand that, changing environmental conditions can alter gene expression. So let's get to the nitty-gritty of this. DNA exists in the cells in the form of chromosomes. So, bacterial chromosomes are circular, eukaryotic, linear. Okay? Something that's not here, I talk about it in the review sessions. In bacteria, you have one chromosome. In eukaryotes, you have multiple chromosomes. So far, so good? Okay. DNA doesn't exist as a naked DNA. It's bound to histones in eukaryotes. Okay. And so-called histone-like proteins in bacteria. So basically, if I ask you, in what form DNA exists, genomic DNA, chromosome in which DNA is bound to proteins. Okay? Understand where extrachromosomal DNA is in eukaryotes and in prokaryotes. Okay, in eukaryotes, mitochondria, chloroplasts; in prokaryotes, plasmids. What about plasmids? Just know that they are non-essential for the existence of the cell. You can remove a plasmid, but they often convey some beneficial traits. So far, so good? Questions? Okay, now we're going to spend some time. We're going to talk about the process of gene expression. We're going to spend some time on this. So, these three statements is basically what you must know about transcription. I just want to break it down, and I mentioned before that in this course you have to learn how to tell a story. Okay, so here's the story. So there's a fragment of DNA called promoter. It doesn't encode anything, it just signals the beginning of the gene. It is before the gene. So the promoter is the place where RNA polymerase binds. Okay? So once the RNA polymerase binds to the promoter, it separates the strands. You can see it right here. Okay? So at the point when RNA polymerase is bound to the promoter and two DNA strands are separated, like this, this is the initiation complex. And then RNA polymerase starts moving, okay, into this direction. I know how to make a little nice arrow here. Okay, moves to the right. Am I making sense? So it moves to the right and it reads one strand Okay, one strand. So this RNA molecule here is synthesized using the complementarity principle, and the template strand is the source of information. Things to remember. Now, if you look at this image, how many strands of the DNA are actually being read? One. It's read from 3 to 5, that's the template strand. Okay. How many strands of DNA do you see? Transcription requires double-stranded DNA. You can't just use one strand. It must be double-stranded, but only one strand is going to be read. How many strands of RNA are synthesized? One. Okay? When RNA polymerase reaches the terminal repeats, it stutters and just falls off. Am I making sense so far? Cool. Now, if we're talking about eukaryotes and some viruses, they are characterized by the process of splicing. So what is splicing? And I'm going to focus on eukaryotes if you don't mind. So this is typical eukaryotic DNA. Okay? You can see that a single gene has 5 exons, which are coding sequences that are separated by 4 introns, non-coding sequences. Am I making sense? So using our cookbook analogy, it's like your recipe for apple pie has fragments of the instructions how to assemble IKEA furniture. Okay, which makes no sense. So you have to remove those fragments before you start cooking. Right? So what happens during splicing? So first of all, this gene is transcribed. So this is your RNA. You can see that this RNA is unsuitable for further reading because it contains that gibberish, the introns, right? So got to remove them. During splicing, introns, these fragments, removed, and exons are stitched together. So now these 5 exons form the actual mRNA that can be used for the protein synthesis. Make sense, everyone? Good? Awesome. Now, sometimes some exons are missing. We call this alternative splicing. You remove one part, it's like, you're making like some fancy recipe, but you don't have potatoes and you just have beef stew, but not potatoes. Potatoes are not included. Still edible, just slightly different recipe. You see what I'm saying? So basically, those are different modifications to the same protein, and those different modifications can have different different functions. Questions so far? All good? So now let's talk about genetic code first. So we're going to start like at the bottom, then we're going to jump to ribosomes and talk about the basics of translation. So proteins have 21 different amino acids. You need to encode each amino acid using nucleotides. Are you with me? So if you consider an amino acid some kind of an object and nucleotides you consider letters, we have an alphabet that consists of 4 letters. If it's DNA, it's A, T, G, and C. If it's RNA, it's A, U, G, and C. Do you follow? All right. So how long should be the words to make at least 21? They have to be 3 letters long. That gives us 64 different combinations. Genetic code. Right here. Okay? Make sense? Now you have 64 words, 21 object to name. You're going to have some excess, right? So yeah, you're going to have the start codon here. You're going to have 3 words that means stop, okay? Whatever, you got the gist, right? You have those. But now you have 60 to name basically the rest 20 amino acids, which means that the code is going to be degenerate, redundant, meaning that, now listen to me very, very carefully, I'm going to say it very, very slowly. It means that a single amino acid can be encoded by more than one codon. Okay? Look at leucine here. Leucine can be encoded by 6 different codons. From the standpoint of human language, and I prefer to use adjectives as an example, if you want to name, to define a mouse to someone who never saw mice, you can tell that person that this is a small, a tiny, a minute animal. A little animal. Does that make sense? Little, tiny, minute, small basically are synonyms. So these 6 different codons are synonyms for the same amino acid. Am I making sense? One of the biggest points of confusion, you have to understand that while an amino acid can be encoded by multiple codons, if you take any codon out of this table, any, I literally mean it, say you take GCU codon here, this codon will always encode alanine. In other words, the word small refers to, well, that size. Not that size, not the size of the wardrobe, right? It's the size of like fist or something. You see what I'm trying to say? So while one amino acid can be encoded by multiple codons, a single codon always encodes a single amino acid. And codon is always 3 nucleotides. Any questions on that part? All good? I want that to sink in. I want it to be processed. I don't want to move on without you basically confirming that, you with me. Okay. I presume everything's good. So, why codons? Like, how does the recognition happen? How does a ribosome recognizes which amino acid to insert? So, look at this illustration here. I wonder if I can do that. Yeah, I can. Okay. Okay. Sorry. So this is mRNA, right? And this is the codon GCC, which encodes GCC alanine. Yes, thank you. Alanine. GCC encodes alanine, right? So this molecule right here, this is transport or transfer RNA. It carries some kind of an amino acid, right? So this particular tRNA that you see on the image has a complementary sequence in its loop, so-called anticodon, that matches the codon. Do you see that? So the codon is GCC, the anticodon is CGG. All good? Right? So, because of the complementarity, they click. Form hydrogen bonds, all right? And it means that whatever amino acid is here, attached here, will be inserted into the growing polypeptide chain. Do you follow? Now, if anticodon is GGC, or CGG, I'm sorry, it is complementary to GCC. GCC encodes alanine. Which amino acid do you think is going to be sitting right here? Alanine. Yes, alanine. Am I making sense? If you would have a codon, I don't know if I can do that. Whatever. ACC, okay, which encodes threonine, then the matching anticodon will be UGG. Right? Do you follow that?
And the amino acid is the letter.
Say again?
The amino acid
No, no, no. The amino acid is an amino acid, a molecule that hangs off, I don't know what's going on here, over here. Okay? So, think of tRNA as some kind of a guide. It provides the connection between an amino acid and the mRNA. You see what I'm saying? So let me ask you this. Imagine that you have a codon, okay, in the mRNA that is CAA. Okay? What's going to be the anti-codon? GUCAA. GUU. Okay? Make sense? So CAA, I'm going to move it away. CAA, what amino acid does it encode? No. What amino acid does it encode?
Okay.
Glutamine. It's glutamine. Okay? It's glutamine. So at the end of that tRNA, there's going to be an amino acid glutamine sitting at this little tail. Does that make sense?
Yeah, I was just wondering, so the amino acids are letters?
No, amino acids, letters are nucleotides. These letters are nucleotides. Those are just a short acronym for an amino acid. Not acronym, it's abbreviation. Okay?
The amino acids would be like the ball.
There will be a molecule hanging off of that stem. Okay? Good? All right. Now, we figured that out. One important thing that I want you to appreciate is the difference between eukaryotic and prokaryotic gene expression. So if you think about, there will be no drawing so far. If you think about prokaryote, okay, I'm going to kind of ignore this because there's a mistake here. It's not DNA. So this is DNA. Can you see that faint blue line? This is a prokaryotic cell, which means there is no nucleus, there's no membrane between the DNA and the cytoplasm. Do you follow? Remember, nucleoid is not surrounded by the membrane. So when this DNA is used for RNA synthesis, okay, that RNA extends straight into the cytoplasm. Making sense? Where it becomes immediately available for ribosomes to initiate a protein synthesis. So the point is, this RNA molecule, this purple one, is still being synthesized. It's not finished yet. Are you with me? But it's already translated. Think of it as you are writing a book and publishing it online on your own personal page, chapter by chapter. So publish chapter 1, start writing chapter 2, but people already read your chapter 1. Book's not finished. You're still in the process, but people have access to it to read it. And think about like 50 years ago when people had to write the entire book, give it to the editor, give it to the publisher, and the whole book comes out, and only then people can read it. So this is eukaryotes. Because first, where is the DNA in eukaryotes? In the nucleus. So you have to transcribe DNA inside the nucleus. Now, if you consider this, sorry, I don't know what's going on. We boned. Oh, okay, I'm an idiot. So if you consider this RNA, is it ready for transcription? No, it has to be spliced. So you have to transcribe it first. You have to splice it. Making sense? You have to get it out of the nucleus in the cytoplasm so the RNA can be translated. So you have a time constraint, splicing, and a space constraint, nuclear membrane. In other words, in eukaryotes, the transcription and translation are strictly separated in time and space. In prokaryotes, transcription and translation happen practically at the same time. Am I making sense so far? Good? Okay, so let's get to this, the protein synthesis story. Now, how does a ribosome knows that the RNA that it's about to translate is actually mRNA? There is so-called, so on bacterial RNA, it's a Scheindel-Gardner sequence. And in eukaryotes, it's a structure located on, so that structure located on 5' end of RNA called cap. Okay, sounds good? Now, for instance, like you may think, oh, this is so boring, like it has no relevance, it's just the interest of some, egg-shaped people with glasses in white coats working on that stuff in the lab. So it turns out when influenza virus infects a cell, it wants to use cellular resources for translating its RNA. Make sense? So it has to redirect ribosomes to its RNA. And influenza virus does it in the most amazing way. What it does, it steals this 5' cap structure from cellular mRNA and attaches it to its own mRNA. So the ribosomes pretty much being tricked, so they bind to viral mRNA instead of cellular ones. The best part of the story is the naming. The process of stealing 5', I'm not kidding, I can find you a manuscript. When the process was discovered, it was named cap snatching. And the enzyme that does cap snatching is called snatchase. It is I listened to that podcast when they described it. So anyway, once, so small subunit recognizes that mRNA and binds to the start codon. Then the first tRNA binds that, complementary C. AUG is the start codon, anti-codon is UAC. So it binds here. And finally, large ribosomal subunit binds to this, and you've got this initiation complex. Sounds good? Okay, so here's what happens. Now, what you're looking at in the center is the snapshot of translation that is caught somewhere in the middle. Okay, so there have been several steps already. Let me try to explain what's going on. So this first place here is called the A, or attachment site. Do you see, I'm going to ask for your confirmation so you follow me through. Do you see that tRNA with an amino acid attached? Okay, so it comes into attachment site. This site in the middle is called a P site, polypeptide. So this tRNA has this long chain of amino acids attached to it. We're good? Okay, now, what happens here is this entire polypeptide chain is transferred onto the incoming amino acid. Now, please answer my question, it's not hard. So, when this polypeptide chain is transferred to the incoming amino acid, this tRNA, does it have any amino acids attached to it after the transfer? No, it's empty. Make sense? Now, this tRNA, does it have just one amino acid Oh yes, it has the entire polypeptide chain on it, plus the amino acid it came into ribosome with. Clear? Next, what happens? This ribosome shifts to the right, so the empty tRNA ends up in so-called E site, gets kicked out. The growing chain turns out to be in a P site again, and A site receives another tRNA. Make sense? Yes? Yes? When we're going to be in the classroom, remind me, I'm going to show it to you using markers. It's very easy. Okay? Just like run the C. Okay, good? And it goes on and on and on and on until ribosome runs into the stop codon, to which the release factor is bound and polypeptide is released. And you've got your polypeptide, and then there's folding, blah, 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 whatever. Okay, makes sense? This is called termination. Pretty straightforward. Does that make sense? Good? All right. So this is your summary. And suddenly I realized that I'm going to have a hell of a time trying to explain what polycistronic RNA is. I will try my best. Okay. So... oh, it records this screen, not this screen. I don't know. I'm not gonna risk it. Anyway, so what are the differences between eukaryotic and prokaryotic transcription, gene expression? So remember, in eukaryotes they are separated. In prokaryotes, they pretty much go in the same time. In eukaryotes, there's an extensive mRNA processing. We talked about splicing, and that's all you need to know. There are other processes, polyadenylation, capping, whatever. In prokaryotes, mRNA processing is not required. Different ribosomes. And this, monocistronic RNA versus polycistronic RNA. So, let me illustrate, well, at least attempt to illustrate, monocistronic RNA. So imagine you have a DNA, right? And you have 3 genes. May I not number them because I will not be able to draw a proper number with this mouse, okay? 1, 2, and 3. So this is DNA. When DNA in eukaryotes is transcribed, you have one RNA for gene 1, separate RNA for gene 2, separate RNA for gene 3. Making sense? When you have the same situation in prokaryotes, this is your DNA. RNA will also contain 3 genes that are not physically on different molecules. It's one long mRNA that contains 3 genes, 3 transcripts. Make sense? So these on the left are monocistronic RNAs. This on the right is polycistronic. Questions on this part? Good? Okay. Oh yeah, now I remember. So now we're going to talk about the best organisms, like this, yes, the best organisms in the world. I still consider them organisms viruses. So we're going to talk about herpes viruses, and I want to apologize in advance. When, I start talking about viruses, I go like a 4-year-old who talks about Transformers. You cannot shut me down, okay? So a lot of stuff that's here, obviously, I'm not asking. What I'm going to do instead, I'm going to highlight what you need to know. I mentioned before, for the most part, you don't need to memorize the numbers, so please don't. I want you to understand this. These viruses are enormous. Okay? They're huge. Largest genome among human viruses is in human cytomegalovirus. Which means they have a lot of genes. And the expression of those genes is tightly controlled in terms of the time. There are genes that are expressed immediately at the beginning of the infection. genes that are expressed a little later, and genes that are expressed late. And if you think about the whole like virus replication thing, it makes perfect sense. Because in the beginning, virus has to, like tegument has to suppress cell immune responses. Yeah, makes sense. Okay, you need some enzymes for gene expression, sure. Later, you need to replicate your DNA, makes sense too. And finally, you need to make a capsid. So herpes viruses have double-stranded DNA genome. This will become important. And they are enveloped. I mentioned before that viruses that are enveloped are not particularly stable. They're flimsy. Herpes viruses are actually flimsy. You can easily destroy them using like alcohol. Okay? Not internally. So another thing that all viruses share is they need receptor to bind to the cell and infect it. Am I making sense? A receptor. Depending on the receptor, the virus may or may not be able to infect the cell. Okay? So these viruses, especially herpes simplex, are sometimes called promiscuous. It's an official term because they can infect, especially in the lab, a huge number of cells. Am I making sense? The receptor that they bind to is pretty common. Okay? So I want you to understand right now a general idea about the virus infection using herpes viruses as an example. So first, they have to bind to the receptor and get the capsid inside. Make sense? Next, they have to release the genome. Here comes the trouble. These viruses have what type of genome? Nope, it's a linear. Huh? Double-stranded DNA. In eukaryotic cell, where is DNA? In the nucleus, not in the cytoplasm. If DNA is in the cytoplasm, it's a problem. Make sense? It means that something's wrong. So to avoid that alarm, herpes simplex tries to transport its DNA into the nucleus as fast as it can. So now it's in the nucleus. It does what any God-fearing nuclear DNA would do. So there's gene expression, okay? Transcription, translation. So eventually capsid is being formed, DNA is replicated, new particles are formed. They travel through the cell, acquire envelope, get released. Sounds good. Nothing complicated. So basically, virus just hides in the nucleus to avoid the detection. And because a lot of proteins that will replicate DNA are there. So far makes sense? What's interesting about herpes viruses, all herpes viruses become latent. I'll explain what latency is in a second. So latency is basically dormancy. Virus doesn't replicate, just sleeps. And the immune system doesn't recognize it. If it tries to reactivate, no air quotes by the way, if it reactivates, immune system shuts it down very quickly. That makes sense? But if immune system is down, like in immunocompromised state, somebody's sick. Okay? Like common cold. Yeah, just sick. Just immune system has other business to attend to. Herpes simplex, for example, can reactivate and can cause a flare-up. Okay, so virus is produced, it is released, it can be transmitted to another person, blah, 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 blah. There are symptoms, and then immune system goes back to normal, shuts it down. So one important thing about latency, which drove me absolutely nuts when I looked at microbiology textbooks that were not free. Pretty much all of them got herpes virus latency wrong, because they stated that during latency, this DNA gets incorporated into human genome. It isn't. It doesn't. Okay? What happens? It exists in so-called episomal state. It becomes circular in the nucleus and stays circular there. In the capsid, in the virus particle, it is linear. But in the nucleus, it circularizes, and it exists separately from human 46 chromosomes. Am I making sense? It's not the part of any of them. Okay. So we're going to discuss three main subfamilies of herpes viruses, and I'm going to highlight the disease, the transmission route, whether or not there's vaccine, and some other features. So, herpes simplex, HSV. V stands for virus. This statement is kind of an old one. Currently, it is acknowledged that probably both of them can cause either. So HSV1 can cause genital herpes, and HSV2 can cause labial herpes or cold sores. You can see that the prevalence of latent herpes by late age is almost 100%. So it just confirms the old saying that, unlike true love, herpes will stay with you forever. And it transmits through bodily fluids. All of them actually are transmitted through bodily fluids. Okay? All herpes viruses are ridiculously infectious. You technically need one particle to establish an infection. But the initial infection looks like cold for the most part. Or just nothing. The biggest problems is this. I just put encephalitis, but herpes virus can cause a variety of other symptoms. Encephalitis probably is the most common because both herpes simplex and varicella zoster, or chickenpox, they become latent in neurons. Okay? So if it's a neuron in one of your facial nerves, right, what's going to happen? It reactivates and gives you a cold sore. If it's a spinal neuron and it's chickenpox, it will reactivate and give you shingles. Make sense? Again, a little inflammation in the skin. It's very painful. Okay?
Yeah, I thought at first, I didn't know that it infect neurons. That tripped me out. I thought it just affect, this epithelial skin cells.
So think of it this way. The question is, surprised that it affects neurons and not skin. It leaves, quote unquote, leaves in neurons. But when it replicates, it infects skin cells and causes those lesions, whether it's a cold sore or shingles. But then the immune system clears it out of the skin, but not out of the neuron, because it's just DNA and it's a nucleus and the immune system has no access there. Did I explain? Excellent. So chickenpox, everybody knows what chickenpox is. The only so far vaccine-preventable herpes virus infection. In immunocompromised can also lead to encephalitis. Both of them are pretty dangerous for newborns if they are transmitted via congenital route vertically. They can be transmitted through placenta. Okay? Especially herpes simplex, it's terrible. Right? Now, cytomegalovirus, I studied it for five years and a change. Bodily fluids transmission, super common, usually during childhood. Kids are not particularly hygienic creatures. In immunocompetent individuals, it causes nothing. Like seriously nothing. So, yes, this was the topic of my research. CMV and cardiovascular disease. I personally think I wasted government money. We did get some legitimate results. But when you have half of the population having CMV, you start asking yourself, is that a risk factor at this point? Or should we just consider it being a part of a background, there are more important factors, such as diet, for example, or lack of exercise. Anyway, there is some legitimate linkage of CMV with atherosclerosis, with high blood pressure, possibly oncogenic properties, but it's just an association, so no causative relationship yet. Here comes the problem. Okay, so extremely severe in AIDS patients, just flamboyant, infects a bunch of different
organs, as you can see. And I didn't list all of it. So from eyes to gut to liver, I didn't include hepatitis, to brain. Actually, CMV is the main cause of rejection not rejection, transplant failure in patients with kidney transplant. Okay? Because they're immunocompromised. Treatable neonatal CMV is horrible. So this is it was a lethal case, fatal, 6 days. These are the deficits and percent of children affected. Children that experience neonatal CMV. It's horrific, okay? Does that make sense? Treatable, by the way, all of them are treatable. Now these two guys, so I'm going to start with Kaposi sarcoma herpes virus actually here. So this virus causes skin cancer in AIDS patients. Okay? Does that make sense? That's it. And I want to emphasize not HIV patients, AIDS patients. You understand the difference, right? So HIV, you can be HIV positive. But if you are on the meds, you don't have AIDS. Okay? But if somebody doesn't take meds, HIV causes AIDS, severe immunocompromised condition, and in this case, AIDS patients, almost all of them have those purple blotches on the skin. It's a skin cancer. Now, Epstein-Barr virus is very cool. It was discovered originally in Africa, and it was eventually linked to these two cancers, nasopharyngeal carcinoma and Burkitt lymphoma. But we know it as a causative agent of infectious mononucleosis. Okay, so if you had mono, you had a BV infection. Okay, it becomes latent in B cells. And when it reactivates, it can cause abnormal proliferation B cells leading to lymphoma, or it can infect epithelia in the nasopharyngeal cavity, causing nasopharyngeal carcinoma. Now, there are a couple cool things, I mean, they're not good, but cool things about EBV. First of all, now it seems that two viruses team up in causing cancers, especially cancers of head and neck. It's Epstein-Barr virus and human papillomavirus. Okay? So, I mentioned that HPV is the most common STD in the United States. HPV can infect oral epithelium as well. So during oral sex, it can be transmitted. Okay? And there's some kind of a synergy between EBV that infects basically nasopharynx, right? And HPV that infects adjacent tissues, and they cause a lot of so-called tumors of head and neck. Am I making sense? Another cool story about EBV, which I really liked. So there was a study that came out a couple years ago, maybe a year ago. So based on the medical records in the VA system, because it's one of the largest and organized same system, same catalog of records in the country. So what they did, they looked at people who had mono ever in their life, okay? And then they looked at the frequency of multiple sclerosis in people who had mono versus people who didn't. And guess what? People who had mono had greater risk to develop multiple sclerosis later in life. Again, the instance of mononucleosis, like when you're a kid or something, doesn't mean that you're going to have MS. It increases the risk. We don't know how, but the association is extremely strong. Make sense? Questions?
Well, because MS is delamination, right?
Demyelination. Huh? Yeah.
Okay.
But it's not in the neurons. So it's latent in B cells.
Oh, that's right.
So we don't really understand. Probably it's some kind of immune system modulation that results in those autoimmune responses. Okay? Good? So now we're going to talk about replications, a few things.
Why we replicate DNA? Because, with the highlighter. Okay. Because we need to pass genetic information to the next generation of cells. Okay? Replication starts at the origin. I want to make a little remark here. One of the points of confusion that I identified over the years. Which process starts where? You should be very clear on that in your study. I saw multiple times when there was a mistake. So, if we talk about transcription, it starts at the promoter. And the enzyme is RNA polymerase. If we talk about translation, it starts at the start codon. And the system that does it is a ribosome. If we talk about replication, it starts at the origin. And the main enzyme is DNA polymerase 3. We'll get there. So far, we good? So, replication starts at the origin. It is rich in AT pairs for obvious reasons. AT pair is weaker and it's easier to separate. So you can see the origin of replication and DNA strands being separated here. Bacterial chromosomes are smaller and there's only one. So, one origin. Eukaryotic, many, because they're much larger and that makes the replication more efficient. Okay? So, new strand is synthesized based on complementarity rule, obviously. And the replication process is a semi-conservative. Okay? What does that mean? Look at these two molecules. Each of them consists of one old strand, old in quotes, old, the strands that came from the previous molecule, and two newly synthesized strands. We good? Questions about the semi-conservative process. You have to separate these two and then copy each strand, each blue strand separately. Okay. Yeah, so why two strands are better? It's a backup. If one's damaged, you have another one to provide backup, then restore the damaged one. Okay, sounds good, everybody? And as we mentioned, replication in eukaryotes is entirely in the nucleus. So not just double-stranded DNA, any DNA, single or double-stranded DNA, is abnormal in the cytoplasm. Yeah, it's not normal. DNA is not supposed to be in the cytoplasm. Good? For now, for like 3 minutes, I'm going to skip this and I'm going to jump here. This is the list of enzymes. You need to know their function. The reason why I made this table is to give you a summary of the functions of replication enzymes. I will 100% ask you to match the enzyme and its function. I can promise you that. Either I will formulate the function and you will have to, figure out what enzyme, or I can tell you what's the function of topoisomerase 4. Okay? Make sense? Okay, so let's go back here. We're going to focus entirely on bacterial DNA. Bacterial DNA is supercoiled, and you need to, I usually compare supercoiling to the wires in the old landlines. You know those phones with the wire, like this spiral thing, it gets all tangled around itself? Supercoiling, pretty much. So bacterial DNA is all tangled, you need to untangle it first. That untangling or removal of supercoiling is done by topoisomerase 2 or DNA gyrase. So here's your DNA gyrase. Once the DNA is straightened up, enzyme called helicase
separates the strands. Okay? So when strands are separated, they kind of, quote-unquote, want to go back together. To prevent them collapsing back into double-stranded structure, single-stranded binding proteins attach to DNA strands and prevent them from collapsing. So now you have a replication fork. Good? Strands are separated. And remember, the image that you see here is just a snapshot of an ongoing process. There were some events in the past. You see what I'm saying? That happened already. And I will have to talk about one of those events. So you've got strands separated, everything's ready. It seems like you can slap DNA polymerase 3 on the DNA and start copying. Not too fast. Let me see if I can do this properly. Okay. I'm trying to make it as big as possible here. Awesome. So now let's draw it. Do you see that little green thing here? This is primer. This is primer, which signals the starting point of replication. This primer is made by the enzyme called primase, and it is an RNA, not DNA molecule. Am I making sense? So again, this primer here, okay, this one is the RNA molecule produced by primase. Okay, we're good? So DNA polymerase starts at this primer, and it starts making the leading strand. Can anyone remind me in which direction is DNA red? 3 to 5. This is your 3, right? This is your 5. So DNA polymerase 3 right here is moving from right to left, reading the blue strand and producing the red strand. Are you aware of any obstacles on its way based on the image? I don't think so. It just goes, right? It just goes. So this is leading strand because the synthesis is continuous, because DNA polymerase 3 is moving in the same direction as does replication fork. Right? Now, copying just one strand is rather ineffective, so you need to copy this one, the bottom one. Can you go in the same, can you put DNA polymerase 3 here and just go in the same direction as the replication fork goes? Why? Yeah, because the direction, right? So you have 3' here and 5' there. So what happens is synthesis of a lagging strand, a sort of saltatory synthesis. So DNA polymerase will move to the right, make a short fragment, then jump back, make a short fragment, and will keep jumping back as the fork is moving to the left of the image. Does that make sense? Each fragment requires a primer, and these short fragments are called Okazaki fragments. Okay? Does that make sense? Now, let's see where we found ourselves so far. We are in the position when you have a leading strand that's synthesized in continuous fashion, okay? No interruptions. You have a lagging strand that is synthesized in the form of Okazaki fragments. So it is, well, fragmented. Are you with me so far? So you don't need to do to leading strand, really. It's good to go. But you got to mend together the pieces of the lagging strand. That's not the only problem that you have there. Which molecule we are essentially making right now in this process of DNA replication? It's a stupid question. Dude. DNA replication. What molecule are we making? DNA. We're making DNA. We're copying DNA. Does that make sense?
These primers, what are they made of? RNA, which is not DNA. We have to replace it. So this is the replacement crew. DNA polymerase 1 removes RNA nucleotides and replaces them with DNA nucleotides. You understand what I'm trying to say? It's like you have a temporary stuff. You remove it and replace it with permanent stuff. Okay? Sounds good?
So both strands are DNA?
Didn't get there, but eventually yes. So your leading strand, okay, I'm gonna try to answer your question. So the leading strand is pretty much the entire thing is DNA, except for that tiny little primer in the beginning. The lagging strand contains, at this point, a lot of RNA primers, right? So how do you remove them? You remove RNA nucleotide, put a DNA nucleotide. Remove RNA nucleotide, put a DNA nucleotide. Okay? What? Go ahead.
And this process is to make, at the end, DNA.
Say it one more time.
I said this process, the ending process, like result, is to make DNA.
Yes, the idea is to get rid of primers. Am I making sense? Okay? You need to get rid of primers. So, DNA polymerase 1 gets rid of primers. The only problem is that when it replaces them with DNA, those Okazaki fragments are still separated chemically. There's no bond between them. And to establish that bond, that's the function of DNA ligase, shown here. So basically it connects, ligates individual Okazaki fragments. Does that make sense? Good? I mean it, like you have to know those enzymes, and I'm going to ask you about this. But a couple more things. I'm going to give you one detail. We're not going to go into mutations now. So if you have if you have a circular chromosome, okay? I'm going to try to draw it, but it's going to suck massively. So imagine that this is one circular chromosome, okay? If you will copy it, at the end, you're going to have a structure that will look like this. So second one, they will be concatenated like like links in a chainmail. You see what I'm saying? Well, like links in a chain, okay? You need to separate them. Separation of those concatenated chromosomes is carried out by topoisomerase 4. Okay? Good? So, second remark that I wanted to make is more of like, I'm going to be pedantic and annoying instructor. What you heard now was a story. I told you the story using that image with the events of the story. This is what you need to aim at. Tell a story. You don't have to tell it without an image. Do it with. Describe what's going on. Am I making sense? Then try to maybe draw it, how I do in reviews, just by yourself, what's going on. If you can do that during the exam, on a piece of paper, that will help you to answer a bunch of questions. Good? All right, folks.